This is our lecture on post-transcriptional modification of messenger RNA. Our objectives are, number one, we're going to look at the nucleosomes and how they're modified to expose DNA that is to be transcribed. And then we're going to look at part of transcription, such as capping of the mRNA, splicing of introns out and joining of exons, alternative splicing, the selection of a number of the exons within different messenger RNAs, RNA editing using guide RNA, and the use of polymerase for tailing the messenger RNA with a number of adenine residues. Let's begin with nucleosome modification. We have looked at it in the past that acetylation, deacetylation, methylation, and demethylation can affect the compact nature of the chromatin. So we understand that in order for transcription to occur, you must relax the chromatin, and the nucleosome is where you have DNA wrapped around the histone proteins. And so histone acetyltransferase will acetylate the histone on the nucleosome, and thus making it less compact and exposing the DNA that can be transcribed. Histone So deacetylase will remove that acetyl group from the lysine residues on histones, and they will make that chromatin more compact, and DNA is not going to be accessible for transcription. Methylation is just the opposite. When you methylate the DNA, you make it more compact. When you demethylate it, less compact, that transcription can occur. And again, here, methylation, demethylation, it can not only occur on lysine and arginine residues of the histone proteins, but it can actually occur on the DNA itself, and it's exactly the same effect, whether methylation occurs on histone proteins or on the DNA that's wrapped around the histones. Methylation will make the chromatin more compact. Demethylation will make it less compact and accessible for transcription. Again, there's a mistake here. So you have inactive genes. That means that this DNA is not accessible, and it's inactive when you have demethylation. So histone deacetylase right here, or methylation. So this should be methyltransferase instead of demethylase. So it should be acetyltransferase to acetylate this, and then demethylase to remove the methyl group. And then this one going back in the opposite direction to make this gene active for transcription. You have to acetylate it, so histone acetyltransferase, that's true, and then this should be histone demethylase. So these two enzymes should be a switch. Demethylase should be here, methyltransferase should be here. Okay? Again, acetylation and demethylation of the chromatin makes it less compact. And again, this is exactly what we're talking about. So you've got repression, gene silencing right here, but when you acetylate it and then or demethylate it, it will become active, and messenger RNA can be synthesized, and the protein then can be made through translation. Histone acetyltransferase, as I said, the acetyl group is transferred to the epsilon amino group of the lysine residue, and this is the acetate or acetyl group right here. That's the epsilon amino group on the R group of lysine. Methylation, on the other hand, occurs on both lysine and arginine, but this is a picture of lysine. So again, on the epsilon group of lysine, it could be non-methylated, mono, di, or trimethylated. Arginine, it could be non-methylated, monomethylated, or dimethylated in two different positions right here. But methylation, again, on these amino acids occur on the R group. Now, let's begin with modification of the messenger RNA. And this is, we begin with capping. You remember, let me take you back to transcription. And I say that the C-terminal domain of the RNA polymerase that synthesizes messenger RNA, as soon as the nascent messenger RNA begins to be shown to come out, right? We say that there was a heptapeptide repeat, that is where phosphorylation, dephosphorylation, especially on serine residues 2 and 5, that is very critical for recruiting groups of enzyme. And as the nascent messenger RNA comes out, the first enzyme, that is enzymes that are recruited, are enzymes that are responsible for capping. And capping involves the following. So you have the NTP N of the messenger RNA. And so the first thing that occurs is that you add GMB to that. Okay, using a phosphohydrolase to form GTP, and this is a very unusual connection because it's 5' to 5' connection right here, and there's a reason for that. It is used for protection. And so after you generate that GTP linkage right here, and then you can methylate it. So methylation occurs in the first two bases. And then the ribose of the second and third base from the 5' end of this messenger RNA. Again, here are the picture. So you can have methylation in the first base, which is G, or methylation in the second base right here, or also methylation in the 2' OH of the second and the third ribose sugars right here. It's the first ribose sugar, second ribose sugar, and third ribose sugar. So methylation in the first two nitrogen bases, methylation in the second and third 2' OH of the ribose. Now let's go to the next one, and that is RNA splicing. So once the messenger RNA is capped at the 5' end, then again phosphorylation, dephosphorylation of the heptapeptide repeat, then remove the capping enzyme, and now the splicing enzyme now comes on to begin to splice the messenger RNA that is being synthesized. Okay. Now, splicing occurs through what is called a spliceosome. And spliceosome is made up of 150 proteins and 5 short or small nuclear RNAs. And there are 5 of these small nuclear RNAs. They're called U1, 2, 4, 5, and 6. And they range in size of 100 to 300 nucleotides. And these are called small nuclear RNAs or snRNAs. And they complex with proteins that are small nuclear ribonuclear proteins. And so these are called the complexing of the RNAs and the ribonuclear proteins. You can call them SNRBs, or it's snRNP. It's a small nuclear RNA and ribonuclear proteins. But for short, we just call those SNRBs right here. All right. And this is basically the way it works. And this is the naked, we're not showing the SNRPs here just to demonstrate what happens. So the basis of the splicing is that at the junction of intron and exon, okay, the beginning of the intron always has a sequence of GU. All right, so the 5' end of each intron is GU, and the 3' end of each intron is A and G. All right. Within a purine-rich region of the intron is what we call a branch point adenine right here. So there is a branch point in a purine-rich region that is not in the center of the intron, but rather close to the 3' end of the intron. And so what happened is you get splicing through two trans-esterification reaction. So the first thing that happens is that the 5' phosphate from the G on the intron now forms a phosphodiester bond between, with the 2' OH. And I don't want you to be confused. Draw it out if you have to. So phosphate group exists in a phosphodiester bond with the 2' OH on the ribose sugar of the branch point adenine. Okay, this breaks this bond, forming a new bond right here. All right, this forms what we call a lariat right here. Lariat is sort of like a lasso right here, and then that's the first trans-esterification reaction that occurs. The second one is that the 3' OH end of that exon then interacts with the 5' end of the next exon right here, forming a phosphodiester bond to break this bond right here, leaving an OH, 3' OH here, and then phosphate forms that phosphodiester bond right here. And so it is kind of confusing, but let's make it very simple. So what we're talking about is this is the 5' end of the exon. Okay, so this is the 5' end of the exon, which is G, and then followed by U. So remember, GUAG. So G right here with its phosphate group interact with the should be 2'. It should be the 2' OH. Yeah, 2' OH of the ribose sugar right here. Okay, and it interacts with the 2' OH of the ribose sugar right here, and so forming a phosphodiester bond right here. And as you can see, the 3' OH of the ribose, same ribose sugar, exists in a phosphodiester bond with the phosphate group of the next base that is here. So the O base is the O intron is from here, and this is the next one, and it just goes on and on. But if we were to cleave these and straighten it out, G, that is the beginning of the intron, and U right here, and this is the branch point right here. And so you can see that there is there are three bonds right here. There's a phosphodiester bond right here. There's a phosphodiester bond right here, and there's a phosphodiester bond right here. So the branch point adenine right here really involves in three of these bonds, and that's very unusual. But this is what forms the lariat right here. Okay, and this is just the same illustration, and that is when you have branch point that forms a phosphodiester bond. This is a lariat, and then this OH then breaks that bond and forms a phosphodiester bond, so that you have a lariat on the left, intron lariat, and the 2 exons that are spliced together right here. Now, how does that happen? Well, as we said, it's mediated by the action of SNRBs, which consist of RNAs, the U1, 2, 4, 5, and 6, and then proteins, ribonucleoproteins, and this unit is called the SNRBs. And so with the RNA and the proteins, they actually mediate the formation of the 2 phosphodiester bond. We won't go into it in detail, but basically the first diester bond that is formed is between the end of the exon and the branch point to form a lariat, and then the second phosphodiester bond that is formed is between the 3' end of the exon with the 5' end of the next exon to form a lariat that is associated with the SNRBs, and now the 2 exons can be spliced together. 2 exons are spliced together. Okay, so this is one that involves the SNRBs, and the same thing can occur without the SNRBs, and that is these are self-splicing. Okay. Self-splicing RNAs right here, where the phosphodiester bond, 2 phosphodiester bonds are formed without the mediation of the SNRB units right here. Again, exactly the same thing happens. OH, the phosphate right here, G, U right here, and AG right here. So G forms a lariat with the 2' OH group right here of the branch point A, and then followed by 3' OH interacting with the 5' of the other exon to form a 2 exons that are spliced together, and then the intron that is now spliced out with the lariat. So this is group 2 self-splicing right here. And the other one is called group 1. Now, the difference between these two is that A is actually a part of that intron, okay? There is, if you can call it that, a branch point G. But as you can see, G is not really in the sequence of that intron right here. It's kind of held in a pocket loosely right here. Its job is to provide an OH group like it did for that, like the A provides for that. But what it provides really, instead of the 2' OH, it actually provides the 3' OH, its own 3' OH, since it's free, because it's not part of this intron right here. So the first thing that happens is this bond is broken. That part of the intron will the G right here, part of the intron, will now form a phosphodiester bond with the G, and now G is going to be at the 5' end of that intron. There's not a lariat form because this G is not part of this. If it were a part of this intron, there will be a lariat right here, but it's not. Okay? And then the next phosphodiester bond, transesterification process occurs between the 3' OH of the first exon with the 5' phosphate of the next exon, and thus forming the 2 exons in one unit right here. Okay? Now, there are a series of slides, and I'm going to skip over them. And this is about alternative splicing. Let me just say it this way. One of the variability that exists in making a number of proteins from the same gene, but making different RNAs, messenger RNAs from the same gene, is through what's called alternative splicing. It could be that one of the messenger RNA may contain exons 1, 2, 3, 4, 5, all spliced together in one messenger RNA. Or another one may contain exons 1, 2, 4, and 5, Another one may contain 1, 2, 3, and 5, right? And so just different combination. But what happened is, instead of making one protein, one size of protein, from a gene that consists of, let's say, 12 exons and 11 introns, it could really end up making from 4, 5, 6, 7, 8, 9, 10 different proteins. And so those are called isoforms. Those are isoforms of the same gene. All right? Different sizes of messenger RNA, and each one will generate different size of protein. They have a great deal of homology, but different sizes, because some proteins include different regions of the larger protein. But it could be that they all serve very similar functions. And this is one way that the cell can actually have a lot of diversity with respect to making a number of proteins from the same gene. So this is called alternative splicing, and that's all we're going to say about it. Please skip over this, and that. And then we come to two other process of modifying messenger RNA. And one is through deamination of cytosine to convert it to U in the messenger RNA. And this is done through an enzyme called deaminase. And then another one is by inserting of a number of U residues in the messenger RNA using what is called a guided RNA. Okay? And again, this is deamination of cytosine to uracil right here. This is, again, just a reaction, deamination of cytosine deamination of adenosine to inosine. So deamination occurs not only for cytosine, but it can occur for adenosine as well. So there are a lot of changes that can occur through deamination. Again, the question that I posed in the previous slide is, how can deamination be lethal? Okay, well, it can be lethal this way. When you have a codon CAA for glutamine that codes for glutamine, if you were to modify that messenger RNA using a deaminase enzyme and you deaminate cytosine to uracil, you're going to convert that codon from CAA to UAA. So you go from a codon that codes for glutamine to a codon that is a stop codon. So what you do is you don't make a large protein at all, but you make a truncated form of that protein. And so this protein may be it's very likely it's not going to be functional because it's half the size of the original protein. And this is the way that it can be lethal by that modification. Okay? And the second one is the use of guide RNA to insert U residues into the messenger RNA. Now, here's the way it happens. And let's put it very simply this way, that you can have a messenger RNA that is made, and you can read the sequence of the gene, and you predict the messenger RNA, and you know exactly what the sequence is, and then you can predict using a program what the protein is going to be and the sequence of amino acids in that protein. But every once in a while, you get fooled because U's are inserted where they don't belong. Now, how does that happen? Well, it can actually lead to variability of the protein product, but here's how it happens. It happens through the use of guide RNA. Guide RNA has two regions. One region is called the anchor, that is used for binding to substrate RNA. And then the other one is called the editing region. The editing region is a region of the guide RNA that forces the substrate RNA that it is hybridizing to insert a U where it doesn't belong. Okay? And here's how it does it. Here's how it does it. Look at this one. So you have an original messenger RNA right here, and you have the guide RNA with the editing region right here. The editing region hybridizes with the target messenger RNA. And look at this. You have GC that pairs up, AU, right? But the guide RNA has two A's right here that don't have anything that are homologous that will bind to it in the substrate RNA. So what is going to happen is it's going to force that substrate RNA to open up and to insert two U's that will be complementary to these two A's. And of course, this A has to be complementary to a base, so it forces that RNA to insert U here, and then these two belong, and then it also forces that RNA to insert U here. So what happened is that you put in two U's, and you put the U's right here, and of course a ligase enzyme will be used to join the final ends together. So the final product after processing is that the messenger RNA has actually 1, 2, 3, 4 extra bases that are used that were not put in there by the transcription of the original gene. They were put in through RNA editing. Okay, and so deamination of cytosine to uracil and the use of guided RNA to insert uracil residues in there. Okay, the last one that I want to talk about about RNA modification is polyadenylation. Now, we also say that again that C-terminal domain recruits capping enzymes first. When capping is finished, then it recruits the splicing enzyme second. When splicing enzymes are finished with their job, then it recruits the polyadenylation enzymes. And these are the last series of enzymes before the RNA is polished and kicked out into the cytoplasm where translation can take place. The complex consists of a number of proteins. There is a cleavage and polyadenylation specific factor. It binds to a region that has the following sequence: AAU, AAA. And then there's a cleavage stimulatory factor that binds to a GU-rich region downstream of this, AAU, AAA. And then there's a cleavage factor 1 and cleavage factor 2, and these are factors that actually bind in between these two sequences right here. Okay, because when the cleavage and polyadenylation specificity factor bind to the AAUAA, and then the stimulatory factor then binds to the GU-rich region downstream, then the RNA loops around so that the two protein complexes interact to form one complex. And then so the cleavage factor will bind in the middle because that's where the cleavage is going to be. And then there's a final one, poly-A polymerase. And that is a protein that will actually cause the cleavage, and the addition of the poly-A tail actually binds. Okay? And so here, let's go all the way here. Now, so you have the messenger RNA that is synthesized, it's been capped, it's been spliced, and now the 3' end is synthesized right here. As soon as these two sequences emerge from the RNA synthesis machinery, then the first series of proteins bind upstream, okay? They bind upstream right here. So CPSF binds to this region right here. CSTF binds to the GU region right here. And then the cleavage factors 1 and 2 bind to this loop right here, because now the RNA is looped around so that these two complexes can interact, and the cleavage factors bind to this area where the cleavage is going to occur, and they direct the binding of the poly-A polymerase, which will bind here, and then it cleaves that area right here. Now, after cleavage, then it begins to add A to that 3' end right here, and you get a series of A right here. Now, to close out, so this is a picture. You're going to have up to 200 A on that poly-A tail right here. Now, let me go back and just review. So you have capping, you have splicing, and you have poly-A tailing. Now, what is the reason for the capping and the poly-A tailing? Well, you can look at it this way. For stability, they make the RNA more stable. They are required to modify the RNA for transport purposes, and then they are also necessary for translation efficiency when the RNA goes out to the cytoplasm and the protein product is synthesized through translation. That concludes our lecture. Thank you very much.